Good afternoon, everyone. It is my great pleasure to introduce Corrie Whisner to give a talk to the CEM, Center for Evolution and Medicine seminar series. Corrie comes to ASU after completing her PhD in Nutrition Sciences at Purdue University and then conducting a postdoc at Cornell with the USDA. So is working for years redacted at the intersection of nutritional physiology, commensal microbial communities, evolutionary theory, and applying this work and insights into actual clinical care and improving directly human lives, particularly at the mother-infant interface, which is, of course, we know, an undervalued and underinvested clinical space in the United States. So with that, please join me in welcoming Professor Whisner.
Thank you so much, Katie, and thanks for the invitation, Anne and Katie, to talk. This is my 12th year at ASU, and it feels like it was a blink of an eye. And so today I'm really excited to present on what we call the Snuggle bug study in our team. This is sort of the first big grant that me and another, at the time, assistant professor got. So for those of you thinking about academia, I try to frame some things about our journey and how we overcame many challenges, and don't be afraid to ask any of those weird questions that you think PIs might not want to answer, because I'm happy to be self-deprecating about my struggles throughout that part of the career, and working towards something that you're truly passionate about and not giving up on the idea just because you get some rough reviews the first time around. So the goal of today is to kind of give you a little snapshot of what sorts of data we have available to analyze so far. So we're still actively cleaning lots of data, and I wish that we had all of our gut microbiome sequences back, but alas, it's probably looking like the end of October. So soon, very soon. And a lot of the Snuggle bug centers around sleep. And if any of you have ever had children or babysat young children like infants, you know that they have some crazy sleep-wake cycles. They don't really have a circadian rhythm. I had a child in the process of doing this study, and this is actually his whole first year of sleep and awake periods. So sleep in the dark and the yellow is awake. And you can see at the center, he's born, and it's just like chaos. And then slowly you start to see these patterns emerge, and these are the types of things that we're trying to study in the Snuggle bug study, and how those relate to risk for rapid weight gain. And I'll talk a little bit more about why rapid weight gain in a minute, but first and foremost, I have to talk about the village, because it takes a village to raise a child, and it certainly takes a village to run a large NIH study. So this is our team. I'm a MPI, so multi-PI, with Megan Petrov. She is the sleep scientist, so if you have very specific sleep questions, I may have to connect you to her, but I'll do my best to try to answer from what I've learned from her. She's also a clinical psychologist by training, and then I'm the nutrition and microbiome person. We have Lilu as our statistician. She's amazing. I just put her on all my projects because I love her so much. And then Elizabeth Reifsnyder was sort of a senior mentor to us. She's a nurse practitioner who's worked in public health and obesity care for a long time. And our preliminary data for this grant were really championed by her, and her allowing us to collect data from her ongoing NIH trials. So she's recently retired, but we still bug her all the time. And then Rosa Krochmalnik-Brown is a senior microbiome mentor. And then these four women, oh my gosh, these are the heart of the project. So Rachel, Karen, Gabby, and Victoria are our project coordinator and our promotoras. And a promotora is a community health worker who really knows the ins and outs of their community, knows how to get in touch with people, find people, connect with them. And so they go and collect all the data and really build rapport and relationships with our participants. And I think because of the work that they do, we have created a community that transcends the research period, and I'll talk a bit about that too. We have clinical collaborators, an OBGYN, Dr. Coonrod at Valleywise Health, and then his colleague in pediatrics, Sumia Panchanathan, and their site coordinator, Maureen, who recently retired also. So we're just taking people into that next phase of life so they can enjoy the world. And then we've got 2 amazing graduate students. Nana just graduated last year from the Edson Nursing and Health Innovation Program. Milena will graduate hopefully this next year. She's in the Exercise and Nutritional Wellness Program. And then Sterling Wright is a postdoc who started with us about a year ago. And so this is not the whole team. We've had lots of master's students working to become dietitians, and then more than 40 undergraduates. So it's really taken an army, and every person's contributions have been so important. And I would be remiss not to acknowledge everybody. Okay, so childhood obesity is a major public health problem. I don't think I have to probably tell anyone that. And you just look around, we hear about it all the time in our health coursework. And while for adults, the prevalence rate has kind of gone stagnant and we're just sort of stuck where we are. It's not really moving up or down. Children is a bit of a different story. There are still some subtle continued increases by certain age groups, particularly some of the teenage years. And then as we get into the younger and younger years, 5 and under, we're still seeing those rates. They're highly variable. You can see for the 2 to 5-year-olds, but this is a phenomenon that's moving into the first 2 years of life too. And we never call a 1 or 0 to 2-year-old obese. We don't have classifications for that, but we can look at patterns that may predict long-term risk. And so when we're looking at infants and toddlers, the World Health Organization has probably the most robust standards for classifying weight status. And so if you're over about the 98th percentile, whether you're looking at the sex-specific weight for length or weight for age metrics, that's basically the cut point where a pediatrician should probably be a little concerned and say, well, let's check in more frequently and kind of look at what you're doing at home. So this is basically just saying that kids are carrying some excess weight for their body size or their age. Now, there's different ways that you can look at that and define risk. So this is just a percentile on the growth chart. But we can also look at how quickly those changes happen, because that's also really important. So here's a standard growth chart. You can right away capture that growth is not linear. It has a curvilinear path over time. And if we were tracking a baby on the 50th percentile, you can see their dots, and then all of a sudden they jump up 0.67 standard deviations to that next line. That is considered rapid weight gain. It's a clinically accepted definition. And if that happens in the first 6 months of life, we know that there are cohort studies that show that those children are more likely to be obese. And if it happens within the first 3 months of life, they're not only more prone to childhood obesity, but also adult obesity and cardiometabolic diseases that often come along with obesity, like heart disease, diabetes. So we see this as a major opportunity for intervening, because what if you could intervene in the first 3 to 6 months of life and then get that baby back on their original growth trajectory and basically set them on a path for life to be free of cardiometabolic diseases. So we're very much thinking on the prevention side versus the treatment side and what we do. And then rapid weight gain hasn't been super well accepted as a clinical metric, although there are clinics that do use it. Oftentimes, people don't always do the best job of constantly going back for all their well visits, especially in the underserved populations. And so probably the most recent systematic review and meta-analysis looking at this in long cohort studies found about 17 different studies that looked at weight trajectories in infancy if rapid weight gain occurred, and then what later outcomes were, whether it's in childhood, adolescence, and all the way into adulthood in some cases. And across all the studies, they found a 3.66 higher odds of overweight or obesity when the infant had that rapid weight gain occurring in infancy. So of course, these studies try to look at some of the predictors that they've collected. Many of these are more electronic medical record sorts of things, so they're limited sometimes in the type of data they can try to associate back. But by and large, what is available in the literature kind of falls into these 4 categories. So there are infant characteristics. If that baby is born at a lower birth weight, they're going to be more likely to have catch-up growth to kind of reach where evolutionarily or genetically they were kind of supposed to be. And that's normal, but then they're going to hit those marks for rapid weight gain, and so a clinician would need to know that catch-up growth is more common in a low birth weight infant. Maternal factors around if mom is entering pregnancy at an elevated BMI, if she also has excessive weight gain throughout her gestational period, those babies are more likely to have rapid weight gain. And then the behaviors that are happening at home. So do parents feed their baby every time they cry? So they're using it as a soothing mechanism, even if baby's not actually showing any cues of being hungry. You may have heard that staring at a television or your phone screen while eating leads to you not maybe being in touch with your cues of satiety, telling you that you're full. That can happen in infants too. And screen time is one of the things that we're also trying to explore because screens are everywhere and we're all on them all the time. And then feeding practices, so formula versus breastfeeding versus mixed feeding. If you introduce solid foods earlier than 4 months, you typically see a greater risk of rapid weight gain. And then if you feed on a schedule versus on demand, based on the infants telling you what they want. In none of these things, sleep is really being talked about. And it is such an important early system that primes every other metabolic system. We have clocks, sleep-related clocks, circadian rhythm clocks, the main one in our brain, but all of our organs have them, including our intestines. And we are, in our group, trying to sort of hypothesize that maybe the intestine helps to prime the central clock in infancy through the timing of when baby's eating, what nutrients are sending cues, and what metabolites are being made. And so that's sort of the next iteration of this, assuming that we can be successful in getting funding, but we'll see. So essentially, though, what we want to do is take a step back and say, well, if you're going to interact with real people, you have to think like real normal people out in the world. What matters to moms? What matters to families? Well, if you download any baby app, these are the things you track: eating, sleeping, and pooping. And you do it on repeat all day, every day, because that's what babies do. And Megan Petrov and I both had babies around the time we were working on this project. This is her little guy, Maxis, and that's my little guy, Sebastian. So we've also just learned a lot from our own personal experiences, for better or worse. So we have that lived experience that we also bring to the project, which I think has made this that much more successful and meaningful to us. So yes, it's an exciting project. So how many of you know a lot about sleep and circadian rhythms? Oh, yay, a few. Okay, cool. So I'm still learning every day too from Megan. So when we were setting out to start this work, we kind of explored, what do we know? How much literature is there really on infancy? And by and large, it's kind of an overlooked area because it's a hot mess and nobody wants to deal with that kind of variability. I guess we're just gluttons for punishment. But here you can kind of see across from birth all the way to 12 months that there's maybe some time points where some important milestones are probably happening. So circadian rhythmicity should be emerging around 4 to 5 weeks post-delivery. And by 3 months, you should really start to see a pattern that looks like this. So it's a cosine wave. And it's not perfect. Like in a perfect world, it would look like this, but that's not what real data look like. So we just want to see that there's kind of that rhythm, and they're starting to maybe consolidate some more sleep to the night, but how much, I don't think anybody really knows the answer to. And what's interesting about all of this is if you think about the reasons that a parent takes their infant to the doctor, you'd think, oh, they're sick. No, it's actually because of sleep problems. That's the number one reason that parents seek medical care for their infant, because they're concerned about their sleep behaviors. And that makes sense. Yes?
What is amplitude of sleep?
So the amplitude is how high these peaks get. So when a baby's starting to develop the rhythm, you'll see it kind of like lower, like this.
What is the y-axis here?
Oh, this is time. And amplitude is in activity, the amplitude is of activity counts. So you're measuring like body movement and how much like vigor there is in that movement. And I'll talk about that later too, and you'll see like some real data. So these infant sleep problems in the literature, we do know that they've been associated with poor child health outcomes, social and emotional difficulties, parental and family health too, because if baby's not sleeping, you're not sleeping, that's frustrating, and you're exhausted. And then adiposity is kind of, although it does make sense, it's not as well studied. So with Liz Reif-Snyder's longer-term cohort that she followed, babies from birth all the way to 3 years, we were able to tack on some of our own questionnaires and collect some fecal samples. And one of the first things we wanted to do is just see what the prevalence of rapid weight gain was and if it related at all to sleep that was self-reported by the parents in the first year of life. So we looked at rapid weight gain at 6 months of age, and then what was happening at 1 month of age in terms of sleep. And we thought that if babies got more sleep, then they would be less likely to have rapid weight gain. And we also thought that if they had more of their sleep consolidating to the night, so moving towards that adult pattern that we hope for, that they would also have a lower odds. And so we saw that if babies napped more than 5 times a day, so we consider napping sleep episodes that happen in more of the light hours of the day, so not at night, that decreased the odds of rapid weight gain. So that would kind of be the opposite of consolidating sleep like we thought. And then then we just looked at the actual proportion of nighttime sleep versus 24 hours. And if a baby had consolidated more of their sleep already at 1 month of age, they were more likely to have rapid weight gain. So this was really interesting. It's sort of the opposite of what we expected to see in the data. And it really told us that there's probably critical time points or windows where these developmental milestones for sleep need to happen, and that when parents are really pushing for their babies to sleep through the night early, may not actually be a good thing. So on top of that, we know really very, very little about the gut microbiome in infancy, and of all the time points of life, it's probably the most complicated to look at because it's a dynamic, rapidly changing system. So this is a little cartoon that represents what the intestinal lining looks like in an infant through childhood all the way up into the elder years. So right away you can see that the morphological structure in the intestine is much more immature. We don't have these deep crypts. We don't have mucus layers that are really thick here. So the microbes are right on top of the cells pretty much, and there's not much of a barrier. Babies also tend to have a leaky gut where there's gaps between the cells. And so what we do know is that we see a lot of acid-producing bacteria in those early phases because acid displaces and harms pathogenic microbes, so it's going to be protective. So acid in this case is a very good thing. And we also don't have a huge diversity because when you have a lot of acid, it actually kind of creates an environment that only acidifying bacteria really like, but it has an evolutionary purpose to protect the cells. And when breast milk oligosaccharides, which are kind of like dietary fiber in breast milk, get fermented by those microbes, they produce things like butyrate and acetate, which are important fuels for the intestinal cells to differentiate and start to build out these crypt structures. So that said, there's a reason that the microbiome composition looks differently. It also, over the course of the first year, really amplifies a number and variety of microbes present. And from this graph that just sort of characterizes some of the major families and classes of bacteria, you can see that there are really robust changes in a few of those groupings. And this isn't meant to be, the be-all, end-all of who's there and what they're doing, but just to give you more of an idea that that first year is super dynamic. And if it doesn't establish itself right, whatever you get at 12 years kind of looks like it continues on. So our goal should be to intervene so that we can get them at the right levels of all the right microbes doing the right things. And in fact, if you look at some of the data on what microbes are present at different time points across infancy, and how that relates to obesity risk. One of the most important things is that bifidobacteria needs to be high at 3 months. When you see it lower, it often is associated with adiposity later on in childhood. And then Staphylococcus aureus has come up quite a bit in the literature as well. So if that's higher between 6 to 12 months, those kiddos are also more likely to have weight issues. And so, of course, those are not the only two microbes present, and many of these studies are based on more, seminal work where we didn't have all these super robust sequencing mechanisms that kind of characterize everything that's there. Some of these are PCR-based where they looked at a very specific microbe for that reason, and that's why this doesn't tell us the whole story. So of course, we were just kind of curious as a group. We got this grant and we're like, we never really sat down and just tried to understand what impact sleep has on the microbiome across the life course, and if there are any key microbial targets that we need to be thinking about that aren't popping up in this infant literature. So over the last few years, we've been working on a systematic review, and I think this is going to get submitted today. Woo! It's been like 3 years, so it's a lot. I keep saying I'm not going to do any more systematic reviews, and then I just keep doing them, and I don't know why, because there's so much work, but it is rewarding. And we found 41 articles, and we only picked articles that kind of viewed the microbiome as the independent variable, sleep as the independent variable, and the microbiome is the outcome variable. But it does have a bidirectional relationship, but writing a systematic review can get really complicated when you're getting into bidirectionality. So we tried to just simplify and looked at things that we were kind of thinking for our own project. The main takeaways are that sleep restriction, disturbances, or misalignment of the circadian rhythm are definitely linked to the microbiome composition, but the overall diversity of the microbes is highly variable. It seems it's study dependent, population dependent, so we're not really sure what's going on there. And for us, the big takeaways are there are only 4 papers in pediatric populations, and only 2 of those include infants and toddlers. So this is like ripe for people to get involved in this space. So I will tell you about those 2 studies because they do set the stage for Snugglebug. So the first is from a group in Switzerland, Schock and colleagues. They're more on the behavioral development and neurophysiology side of things, but they did happen to have microbiome and sleep measures. And so you'll notice they have this weird variable called sleep day. This is an amalgamated variable that they created, which accounts for duration, frequency, and regularity across a week. So how regular the amount of sleep is across the week. And then they looked at alpha diversity, which is within sample richness. So how many unique features are in that microbiome. And they looked at 3, 6, and 12 months. And you can see that we've got the 3 and the 6 month data in the gold and the blue. And there's sort of this relationship that shows the more sleep that you're getting in the day, the lower the diversity. And now in adults, we want high diversity, but early in infancy, lower diversity is better. When we think back to that earlier picture where we want those just very few acid-producing bacteria. And what I take away from this too is that the first half of infancy, that relationship is there, and then it dissipates by 12 months. So again, our intervention area should be in the first half of infancy. The other paper that ended up in our review is from our own group. So this is based on some of that preliminary data that we generated from Elizabeth Reif-Snyder's ongoing trial. And so she had 36 kids left to follow up with at 3 years of age, and she had been following them since they were born. And we're like, hey, can we get poop from your 3-year-olds? And she's like, sure. So the participants and her team shipped their poop to us at ASU, and so we were able to go back to sleep surveys during infancy and then relate that to the microbiome of these kiddos once they turned 3. We looked at the proportion of night to day sleep and then total sleep time. And for those not familiar with the microbiome, we looked at alpha and beta diversity. Alpha is within the sample. How rich or evenly distributed is the community? So here we've got 1 species of flower, here 3, and there 2. And then beta diversity is when you compare across or between samples. So these 2 bouquets have no overlap at all. So they would cluster away from each other in multidimensional space. And if we were looking at these 2, they would have a little bit of overlap because they both share tulips. So that's how we kind of look at microbiome when you have like hundreds or even thousands of features. It gets a little tricky to analyze that data. Here is at the 1-month mark, looking at what happened by parent report baby sleep and looking at the microbiome. So we saw that babies who had a lower proportion of their sleep in the nighttime had a richer microbiome. It was more evenly distributed. And then when we looked at between samples, there were differences emerging based on abundance of the microbes present and also their phylogenetic diversity. We looked at this also at 6 months, and here I'm just showing you Faith's PD. So this is an alpha diversity within sample metric looking at the phylogenetic composition of the microbes. And we have total sleep time here, and then the proportion of nighttime sleep here. And if babies were getting more than 13 hours of sleep, so we just used the median splits because it was such a small sample of 36, and then less than 66% of that sleep was happening in the night, they had a higher phylogenetic diversity. And then because we know other factors play a role, we tried to squeeze as many variables into this model as we could. So we did stepwise regression to kind of allow it to kick some things out. We did include feeding, sleep duration, the proportion of nighttime sleep, and then the change in weight for age z-score. And so the only 2 variables retained were the proportion of nighttime sleep, which was statistically significant, and then weight for age z-score was trending. So these are all data that we kind of used as preliminary data for the grant. And with this and the other study, that was finally the last piece that we needed to get across the finish line because we kept getting, this is too risky. You might not find what you think you're going to find. We need to see that other people have done this. And we're like, oh. They always say that NIH is high risk, high reward, but it's not. It's very incremental. And the main takeaway with this is that alpha diversity was lower in toddlers who obtained more of their total sleep at night and less sleep overall at 6 months. And then beta diversity, the main takeaway here is if you look at all these different metrics and ways that we can slice and dice the microbiome by presence and absence, phylogeny or not, and then abundance, they're separating based on the proportion of sleep that they're getting at night. So there's something going on in infancy with consolidation to the night for sleep and how the microbiome looks several years later. So that leads us to our very first R01 and probably helping us get tenure because that's hard to do. So we were very excited, like the minions that you see here. And the National Heart, Lung, and Blood Institute is the institute that funded us. If you ever do sleep work, this is the institute that funds the vast majority of sleep work. So we picked that for that reason, and also it had the best early stage investigator pay bump. So your score could be lower and you still would get funded over someone else being a more junior investigator. It's still a good project. I'm not trying to stab ourselves in the back here. So the goal of our study was to look at how sleep-wake patterns, the gut microbiome, and feeding patterns develop and evolve over that first year of life and how it affected the risk for rapid weight gain. So we recruited mamas in the 3rd trimester all the way up to 3 weeks postpartum, at which time we would screen and consent. The purpose of going pre-delivery was so that we could start to build rapport and connections with mothers who might be interested. And then we longitudinally followed up 6 times in the first year. So 3 weeks, 8 weeks, 3, 6, 9, and 12 months. We measured sleep, both subjectively and objectively. We got dirty diapers, which every mom is totally happy to give away. If mom was breastfeeding, we said, hey, if you're willing, we'd love to have a breast milk sample. Same for urine. It's really hard to collect baby urine samples. But we did get quite a few, I think like almost 600 over the course of all these time points, and then tons of surveys, so many surveys. We asked about nutrition, family dynamics, you name it, we probably asked about it. This is our primary outcome, rapid weight gain at 6 months, and then again, we want to know if anyone rescues back onto their original path or ends up having a weight for age z-score greater than the 85th percentile by 12 months of age. We recruited 205 mom-baby pairs. They were generally healthy. Mom couldn't really have any major cardiometabolic conditions, with the exception of we did allow gestational diabetes. Mom couldn't have been using illicit drugs during her pregnancy. The baby had to be born normal weight, so between 2,500 and 4,000 grams. And they had to be discharged from the hospital together because we wanted them to be able to feed baby as they had intended. And if you're not discharged together and you're trying to breastfeed, that can really mess up that early establishment of feeding. And these are all singletons, so no twins, and they had to be born full term, and we defined full term as greater than 37 weeks gestation. We used a cells to society framework to guide our questions and ensure that we were collecting robust data for covariates. And so here you can sort of get an idea of the different types of data that we were collecting. And this is basically the social ecological model, but we added in biology right here, hence the cells to society framework. And then we are looking at aspects of the child, mother-child interactions, the broader family. We have a very unique demographic in the Phoenix metro area, so knowing that people are immigrants or coming from Hispanic culture, we do ask about acculturation, what their parenting styles and beliefs are, all these things that can really impact behaviors and biology. And then we all go all the way out to the broader community level, so are they using any subsistence programs like WIC or SNAP, what their economic status is, does the baby go to daycare, what other exposures will they have essentially is what we're interested in. And for sleep, to measure that objectively beyond what had kind of been done previously with more survey-based work, we used the actigraph from AMI. And so you can see a picture of what it looks like. It's like a digital watch from the 1980s. We pilot tested this. Moms did not want to put this on their baby. One, when they did, it had screws sticking out of the back of the watch face, and the band is really rubbery, and baby skin is so delicate, it would just kind of rub their skin off. So we quickly had to get creative. So we worked with a local seamstress who made us these awesome soft neoprene pouches so that we could remove the watch band and then embed the watch face in a little pocket that has a clear plastic cover, so it's kind of protected from spit up and urine and poop. And this is a light sensor here so that we actually can see what the light exposure is during periods of activity or sleep, and then the parents hit a little mode button to say, okay, I laid baby down for a nap, and so we kind of know to cue in to look for when their activity level changes for sleep. And then the other thing on the back is there's actually a temperature monitor because your body temperature changes when you sleep. And so we pilot tested this quickly, and we were still getting all those types of data that we needed, and it seemed valid, so we're like, yes. And we had all these fun, cute patterns, so moms could pick and choose which one they wanted, and this worked. They still kind of look like they're on house arrest, though, which I think is adorable. So once you get actigraphy data back, it basically collects body movements in 1 minute epochs or epochs, depending on if you want the British wording or the English wording. And then you can get all kinds of different metrics. This is just a short list here, but total sleep time, wake after sleep onset. So how many times do you wake up after you fall asleep? We all have little micro awakenings in the night, and sometimes we don't even realize they're happening. Sleep onset latency is you lay down, but you don't fall asleep immediately. So what's that time that it takes you to fall asleep? And then the longest nocturnal bout, how many naps, all kinds of things that might be important for understanding how tired you are when you go to sleep, and then how robust that sleep is. For the microbiome, so I told you we used diapers. The moms would swab the diaper with sterile swabs and then give us both the dirty diaper and the swabs. We saved the diapers to archive samples for other uses, and then also have it as a backup in case our 2 swabs failed, which only happened less than 1% of the time. So that's great because we can put that in future grants to be like, yeah, we know what we're doing. And then all of the diapers come back, we process them, we extract the microbial DNA from the swabs using proprietary manufactured kits. We don't really try to make up our own reagents and things. These companies, they do it well. And so it's just better when you're like, you guarantee this works. And so we're going to do that. And then we had originally only been able to budget for 16S sequencing. And in the process of doing this project, long read sequencing became available so that now we can get species level rather than just genus level information. So we swapped that out. And then we are archiving samples for deeper sequencing later. So our sequencing is currently at the sequencing core now. Hopefully it is getting sequenced at this moment, I hope, fingers crossed. And then we'll have, I think we sent 1,005 samples, so that's pretty cool. And then I just want to take a minute to just talk about our participant engagement, because this is something that I think we're all very proud of. And we had a really rough time because we started this project in the early part of the pandemic when there was a lot of uncertainty. People had lost their jobs, and then babies weren't actually eligible for vaccination. So guess what? We designed a study to go into people's homes, and they did not really want us in their homes. So we had to pivot and get creative. We did curbside drop-off. We made all kinds of instructional videos. We'd Zoom chat with people and walk them through the measurements. And then once things kind of settled and babies were vaccinated and there was a little less fear and concern and people trusted us, we started to host biannual picnics in the park to further engage with our wonderful participants. We did story time. We had big mats with toys everywhere, and the big brothers and sisters would come and play. We did raffles with like handmade goodies. We call that one Hawaiian barbecue. We made little baby teethers. Everyone got a little goodie bag at the visits with sometimes homemade items, always diapers and wipes because those are expensive and useful. And then we would have a photographer come and take professional photos of the families at the park. And so these are some of the pictures you're seeing where participants have allowed us to share. So these are really things that a lot of participants in our exit survey talk about how like meaningful those were, and they didn't just feel like a number in a study, and that they felt like a person who belonged to a community. And then the last piece, and this is really our big thank you to them, is a story of a snuggle bug. We call it the baby data book. One, it keeps us accountable to keep up with cleaning data in real time, but we basically take snippets of all of the dyad's data and we put it into this really cute book, and then we print it and cut it and like bevel the edges, and then we mail it to them. And so these are really nice keepsakes that everybody gets. So just to tell you a little bit about our families, we had a very diverse group. So a quarter of our moms were first-generation immigrants, and a third were enrolled in WIC. So that's a government assistance program that provides supplemental food if you meet a certain threshold for your income, basically kind of below 98% of the poverty line. And then we hit 36% Hispanic immersion for our project, which is a little better than the last census in 2020. So we were really proud of that we were actually like reached a more diverse cohort than the local census area. And then another 11% were non-Hispanic, other racial groups. So we did a good job of kind of mirroring the population in which we live. Our moms were on average around 32. Many of our snuggle bugs had siblings, but 40% of our participants were actually first-time moms. So how cool is that to be a part of this like amazing first-time experience in your life? And then we get to be like flies on the wall and be a part of that process. It was really rewarding. Most were vaginal births. Many of our snuggle bugs have pets in the home, and we wanted to ask about that because it really does change your microbiome when animals and babies are licking everything and putting stuff in their mouths. And then we hoped for 50-50 male and female babies, but for whatever reason, we tended to have a few more girls than boys. They have like the most average weight. that you can imagine. Solid foods were introduced around 5.5-ish months, and just thinking about impact beyond what we write in grants of like, we're going to affect change and it's going to make policy and clinical care better. We made all those videos to help our participants collect data, and actually, I mean, this is viral for us. I don't know, almost 500,000 views for our urine collection bag. It is not a good video. But people were like, thank you so much. I had no idea how to put this bag on my baby. They just gave it to me and said, collect urine. So we taught them how to put a pediatric urine bag on under the diaper without it all spilling out everywhere, which happens a lot. So that was really rewarding also, just to see that our tools were useful beyond the study. One of the most important things we wanted to look at was how our babies were being fed. And so this is really cool. Like half were exclusively breastfed, and many of them sustained that for the full 12 months, which is super great. These are our mixed feeders. They're getting breast milk and formula, and you can see that a lot of them start off, this is actually a common Hispanic practice, to give formula to supplement early on until they kind of see if their breast milk comes in, and then they're switching up here to formula or down here to exclusive breastfeeding. And then up at the top, those are our exclusive formula feeders. The average duration of breastfeeding was almost 8 months. And so depending on which racial and ethnic group you're looking at, this is slightly better than all of the racial and ethnic demographics. So we were pretty excited about that. And this is one of the things that we would kind of share with our participants in their baby data book is, what's your baby's favorite food? And then we just clumped it all together in a word cloud for presentations. They loved bananas and eggs and yogurt, cheese, and oatmeal. And we looked at a couple of our screen time and sleep characteristics. So some of our babies by 6 months were already being set in front of screens, and we were finding that those babies get less sleep and had later bedtimes, but the quality of their sleep wasn't affected, which is interesting because we always talk about blue light exposure and how that can impact the quality of your sleep. The babies didn't seem to be affected by that. And then maternal stress, that's a very important thing that we have to think about, and we screened for it, and we helped give resource lists and connect people to the right resources if they were having any depression, anxiety, or immense feelings of stress. If a mom reported higher stress, their babies woke up more in the night, which is interesting. So it's sort of a chicken and egg question because many of the parents are also room sharing or bed sharing. And so are they disrupting the baby by waking up, or is baby in tune with mom's cues and knowing that she's stressed? And then moms who fell asleep later at night had a harder time getting rid of their pregnancy weight, which kind of makes sense. With regard to the microbiome, this is just from the preliminary data we have. These are sort of the top 10 microbes that we see. So you see bifidobacteria and lactobacillus a lot in there. So those are our acid producers. And we were super excited to see that in all the samples that we preliminarily sequenced, every single baby at least one time point had Bifidobacterium infantis. This particular microbe, some people feel is like disappearing. It's definitely more prominent in babies who have been breastfed, but we were happy to see that at least in this early subset of sequencing, that our babies were at least expressing it. It's a highly beneficial microbe. I'll just talk through a couple of the analyses that our students and trainees have been working on because I really want to highlight the hard work that they're doing. So Melena's really interested in how feeding practices and bed sharing influence different health behaviors in early life. And so she used some sleep data from our subjective surveys, so the Brief Infant Sleep Questionnaire across up to 6 time points if we had it all, and then ran a multi-level growth model to look at nighttime sleep duration as her outcome. And then she looked at bed sharing, infant feeding, and maternal education as a covariate to account for the fact that lower educated mamas tended to be the ones who dropped out from the study. And what she found was that as someone increased, if we treat the feeding variable ordinally, so you're moving from exclusive formula feeding to mixed feeding to exclusive breastfeeding, each of those increases led to almost an hour extra of sleep a night. And you can sort of see how our breastfeeders in green and the blue are the non-breastfed, that is the case up through 6 months, and then they kind of even out there at the end. So again, that early half of infancy might be really important because that effect is weakening over time. And there's a lot of, I guess, testimonials out there that moms will follow where they say, well, if you give your baby formula right before they go to sleep at night, they're going to sleep through the night. And our data are actually showing that breastfeeding is leading to more sleep at night. So this is going to be really important for trying to change those beliefs in the community. And then Sterling has been really interested in looking at the microbiome. So he used what we had in terms of actual available actigraphy data. So it takes forever to score those actigrams. And he looked at alpha diversity and found no impact of feeding mode on alpha diversity. But he did see some differences for beta diversity. So, and he also accounted for several covariates with that. And because beta diversity assessments just kind of tell you, yes, the community structure is different, but you don't really know what is different. He did differential abundance analysis, and he found that Lactococcus lactis was enriched in the exclusively breastfed infants compared to both the mixed-fed and formula-fed. So there, even when you're getting any breast milk, Lactococcus lactis was lower. This is a good fermenter of many things, and there were a couple trending bifidobacterium species as well, but they didn't achieve statistical significance.
When you measure beta diversity, is that just like 3 months versus 12 months, or is it every pair?
He plugged in all the time points that we had and then adjusted by participant, because then you're kind of repeating the same person in the model. And then Sterling also looked at day to night sleep ratio in relation to the microbiome. And again, it seems like that proportion of nighttime sleep is a really important variable. It's emerging in like several of the analyses that we're looking at. And here he ran a model with that, accounting for the individuality of the participants, and then nesting each of the participants or an interaction, with nighttime sleep. And so again, he's seeing a significant association between the nighttime sleep proportion and the community structure. So there are significant differences. So again, he did differential abundance analysis to look at that in relation to the nighttime sleep. And those time points or infants who had a lower proportion of their nighttime sleep tended to have Anaeroglobus massiliensis and Enterobacter cloacae, and then Haemophilus D species. We don't know much about the first one. The second one has been linked to obesity, so that's worth exploring. And Haemophilus D is an opportunistic pathogen, whereas if those infants had greater nighttime sleep, then they had this awesome short-chain fatty acid producer, Mediterraneibacter faeces. None of the Q values, which are the adjusted P values, are statistically significant here, so this is all very exploratory. But it gives us some ideas of microbes that we might want to explore a little deeper and then also revisit once we have our full data set. And then lastly, I'll just take some time to talk about circadian rhythm activity and how it affects or is associated with rapid weight gain risk. And this is our very first aim of the project. This is underpowered, so just be prepared to be underwhelmed. No. So before we talk about that, though, I just want to quickly go over some metrics of sleep here. So our babies wore ankle actigraphs for 5 consecutive days to the best of their abilities, so that we would hopefully get 3 full days of data, because they think these things fall off, babies poop on them, and then everything has to be washed, and mom forgets to put it back on. We piloted a lot of this stuff on me, and I was like, I can't do this, Megan. I'm a discombobulated mess. We've got to rethink some of these things. And she's like, but you know it's important, Corrie. And I'm like, this is real life. I don't know why we propose these crazy things. So here we've got this nice little circadian curve. And so Again, this is time, and then these are activity counts, so measuring babies' movements. And then you can look at periods from peak to peak. You can look at magnitude, which is also called the amplitude. And then the goodness of fit, and we're going to talk about these two, amplitude and goodness of fit. Goodness of fit is how well does the full data set for that baby actually fit a cosine wave. And then there's non-parametric ways that you can do this too. And those may be more appropriate in the early time points where babies don't actually have a full circadian rhythm yet. If you look at babies' data, what a real actogram looks like, this is a baby at 2 to 3 weeks after they're born, the 6 weeks and 12 weeks. So you see that the amplitudes are increasing across these three panels. So the peaks, how robust that activity is. And you up top, it's kind of all over the place. These are the nighttime panels and these are the daytime panels. And then essentially what you want to see over time is that the amplitude increases. So here is 3 months, 6, 9, and 12 months. So the peak is rising over time. So our awesome statistician, Lee, did this not fully powered analysis, take it with a grain of salt, looking at visit-to-visit changes in circadian rest activity rhythm metrics and the relationship to rapid weight gain. And she used time-varying effect models, which are a better fit for nonlinear aspects of growth and developmental patterns. And so she ran several different models. The first model looks at the overall sample mean, and then model B looks at the between and within person effects. And so here are these really hard to interpret graphs that I'm now going to wave my arms and tell you what to believe. On this side are our sample level mean ones. These are the between person and within person. The x-axis is time, so over that course of the first year of life, and the y-axis is the beta coefficient between the circadian rhythm metric and the rapid weight gain. And essentially what we are seeing is that you kind of see these 95% confidence intervals approaching 0. That's a good thing. We want to see that. So I've circled those graphs that are kind of starting to show in that first 6 months. We're seeing that. We're moving towards the association that we hope to see. And now I'm going to attempt to explain this to you. And I'm just going to scroll to my notes really quickly so I don't misspeak. Okay, so over infancy, regardless of time point, greater person-centered mean amplitude for the baby is trending toward an association of not having rapid weight gain. And then over here on the right, this is showing that greater within person variability from a person's own mean amplitude across infancy had a trending association with not having rapid weight gain. So what the heck does that mean? Well, let's say we've got 2 babies. They both have a mean amplitude of 75 activity units. And then we look at all of their values over the full first year. And one baby starts at, let's say, 25. And then at 12 months, they end up at like 125 activity counts. So they have a much bigger spread. So they're going to be less likely to have rapid weight gain than the baby who also had a mean of 75 activity units, but started at 50 and ended at 90. So they're not really increasing their amplitude as much over the full 12 months. So we really want to see those peaks bouncing up over time. And if they don't, they're at greater risk of rapid weight gain. We looked at the same thing in relation to the fit for R squared. So how well do the data fit a cosine curve? And we saw the same thing. Our confidence intervals are approaching significance, but they're not quite there yet. And we have a lot of work to do to finish up scoring our last actograms. We've got about 20 participants left to complete. I only know because I am the one that prints all of the baby data books and cuts them. So that's how I keep track of where we are with sleep data. So we're getting so close. And to bring this all together, we're seeing essentially that breast milk exposure seems to be important. It's associated with more nighttime sleep, and that effect is most potent in early infancy. We're seeing that infant feeding and sleep have an impact on the gut microbiome, or at least are associated. I don't want to make any causal statements. That sounded a little too causal. And that newborn period, first month, second month, third month, those are the times where I think we really need to understand what's happening, because those are where we're seeing that parents want to consolidate nighttime sleep, but maybe it's too early developmentally. And we don't want babies to adapt and have a too early or too rich microbiome. And that we're seeing in at least our preliminary stuff from the toddlers that this stuff carries through over multiple years. So what is the long-term impact of these events happening in infancy? And then exclusive breastfeeding led to a different microbiome compared to even mixed fed in the formula-fed babies, which I was really hoping those mixed feeders would still be in there, along with, so that basically any breast milk exposures leading to a different microbiome. But it's such a contentious topic, breastfeeding. And to me, a fed baby is the best baby, but there are some strong opinions about breastfeeding out there. And I do think it's great, but at the end of the day, a baby needs to be fed. And then lastly, infant sleep. Hopefully, once we get our full data set, we'll see those trends and maybe those 95% confidence intervals drop below 0. But basically, frequent napping seems to be important in early infancy. So let that baby sleep when they're tired. Sleeping more at night during that early phase is maybe not necessarily a good thing, and it continues to affect the microbiome into toddlerhood. And then consistent, robust rest activity rhythmicity is also important in relation to rapid weight gain, and TBD on if our full data set tell the same story. So what does all this mean? Well, we've got so many covariates we haven't even been able to try to account for yet, and we have plans for that. We've got some ideas of what we might be seeing, but I'll just sum all of this up in that clearly timing is key, and there are windows of opportunity, and the levers that we pull during those different windows of opportunity may differ by person, so a personalized approach, but it also may need to depend on the developmental stage and where that baby's at and what other factors are interacting. And so we will have this amazing data set to be able to explore a lot of these really cool questions, and hopefully be able to improve infant health for the long term and can do our part to contribute to lowering the obesity epidemic. But with that, I will leave you with some more Snugglebug love and be happy to take any questions.
All right, we're gonna start. There was a question that somebody that's watching from Zoom submitted earlier in the talk. Great, thank you so much for that wonderful, wonderful talk. The question, when you were talking about that, soothing the baby with food, either breastfeeding or formula feeding or other kinds of foods, that was predictive of rapid weight gain. Mm-hmm. And so the question here, or the topic that the questioner would like you to speak to, is that it is interesting that feeding for soothing is a problem today, because that is how it probably was during Paleolithic conditions. But perhaps the difference is that while the hunter-gatherer moms feed the baby for very short periods to soothe in response to any fussing, this may be different to kind of longer periods of consumption in the contemporary setting that you're studying. What are your thoughts about why would feeding for soothing be risky, given that kind of behavioral interaction was likely quite adaptive?
That's a really good question. And I'm not sure that the existing literature that shows that relationship asks about how long babies are being fed, and that's probably a really critical part to that. Most of the studies have a survey, and the mom is asked to check all of the things that they do to help their baby get back to bed, and it's literally, I feed my baby, and that's it. So that's all we really know. And so maybe in the future we do need to ask how long, do you change first and then give a little milk? Do you give a little milk just to calm them and then, check diaper and then lay them back down? And I honestly think there's probably other interacting factors there that aren't being accounted in those models too.
Fantastic. Are there any, yes?
Great talk, thank you. I actually have two questions. One is related to your preliminary study, and you looked at the microbiome in 3-year-olds, and I was really curious whether you had any data about whether those kids, that's right about when kids start becoming lactose intolerant, if they're going to be. And so I was curious whether or not you knew or queried people about that. And then my second question related to this study is these are, I'm assuming here in Phoenix, so urban population. Has there been much work done in rural babies, and do you see the same kind of microbial communities? Because we do see an urban, rural, or a weird traditional divide in terms of microbiomes, and so I was curious how that looked in babies.
Yeah, both very good questions. I don't think that the parent study that Liz did asked about lactose intolerance. There are diet records, so assuming that the data are of high quality, which diet data is just sometimes a hot mess. You could probably go back and look at it. Are they getting milk or not? And then compare there, but you wouldn't necessarily know if they're reporting lactose intolerance. We ask about allergies in our study, and we didn't have anyone report that specifically. So it's something that we could maybe explore in our project.
My daughter became
Oh,
lactose, so I just remember that.
yeah.
So I was kind of curious if that she starts shifting the microbial.
Yeah, well, and it definitely could, for sure. And I don't know what the implications of that would be, and especially in this time right now where plant milks are like busting at the seams. I feel like it used to be the orange juice aisle at the store, and now it's the plant milk aisle at the store. And what that means, because those milks are not compatible or comparable to cow's milk. And then your other question was about urban versus rural. Yeah, most of our participants are from the Phoenix metro area. We did drive all the way out to Cave Creek a few times, but that's not very rural. And I'm not aware of any studies that have specifically looked in rural communities for these types of questions. And you're right, that it's really important to have that comparator, different microbial exposures and different behaviors too, different lifestyle.
Follow-up question in the Zoom chat. Where you found statistical significance, given that this is preliminary data, what were the effect sizes, and do you interpret them as being biologically meaningful?
Who is that person? These types of the microbiome stuff, the way the models work, the effect sizes are actually bigger than you'd expect. But when you're talking about feeding and diet, they're small, but they're meaningful. And over time, I think the magnitude of their effect grows. And I don't have an answer to, the exact biological plausibility of how, strong that would be. And I don't want to say that what we have now is yes, of course. We just need our full data set, I think, to really know.
Yeah, fair. I often remind myself that eyes were initially a mutation for just light-sensitive patches of skin. So it can be very small, subtle differences that can have, long-term evolutionary trajectories. Any other questions in the room? All right. Well, if we could all join me again in thanking Dr. Whisner for coming and talking to us.